One of the things, April, you'd hit on in a previous answer as well is thinking about sort of the cost of goods, right? You were watching some of the technologies in terms of new capping, right? Technologies, ways to reduce the amount of wasted capping, right? Reagent. So managing cost of goods is such a huge issue. And so I want to pick each of your brains in closing here today. Like, what are some of the greatest, and I would argue most manageable, right? Like, we all have pie in the sky hopes and dreams, but where are you seeing some of the most meaningful improvements we can make today in terms of innovations or technological improvements that you think are going to improve the cost effectiveness of our mRNA-LNP manufacturing processes? So Adi, let's start with you, and then we'll go to Tyler, and we'll end with April.
I believe April already touched on this because she did mention the cost associated with these proprietary capping reagents. Now, they are a major hurdle. So if we can somehow reduce that burden, that'll be really helpful. And on top of that, the problem is, for a normal IVT reaction, it's required in a high concentration, but it's not completely utilized. So basically, it's the most important cost factor. At the same time, after the process is done, it's just waste. So if you could somehow reuse it, so that's an approach that we are taking here, trying to somehow recycle the capping reagent that has not been incorporated in the first IVT to, for subsequent IVTs, right? So that's one thing. The other option, again, she mentioned use of enzymes that don't require such high concentration of the capping reagent. Again, a very good solution. But the problem there again is these enzymes are also proprietary. So, you're teaching one proprietary thing for another. So I am still thinking about this from a process engineering point of view. How best can I optimize the process, with whatever is available, rather than being overly dependent on something proprietary. So that would be the approach that I would take. So in order to do that, again, that's one thing, recycling of unutilized substrate or capping reagent. The other thing, as far as the other major cost driver, would be the downstream operation. So there, we also are developing processes that increase your resin utilization for purification of RNA. So all of those things. So I view this from a process optimization point of view.
Yeah, absolutely. Tyler, what are your thoughts?
Yeah, Adi hit on a lot of those, and I would reiterate what April said. I think utilization of novel enzymes, yes, they come with more costs, but if they can drive your efficiencies on the upstream side and reduce impurities, I think that's one place that we've definitely focused. And I think in general, focusing on optimizing your yields on the upstream while reducing impurities is going to also have a profound effect on the downstream process and really allow for the downstream to be much more efficient with yields. And so I think really coming back to Adi's point is if we can make the upstream more efficient, have less junk in there, then our downstream will also be there. And I think these engineered enzymes have enabled us to really limit our downstream needs, really not having to go through a lot of different chromatography steps, and can get some pretty clean product out of it. So those are the two places. And then, the economics of scale, really just getting to larger scale batches. There's so much interest from raw material suppliers to make these things cheaper. And so I think as we start to need more, prices will go down as well.
Awesome. April, you're the woman of the hour here. They've both referenced things you've already said. So. Do you have anything else to add?
The couple of things I'd add, like thinking more strategically about this, and I am starting to see a little bit of movement, is consolidation. So it would be nice to be in a place where you can go to a CDMO and just get everything you need for the mRNA production from that CDMO. So Aldevron's made a deal with TriLink. They can now source the cap directly, which is great. That makes life, and others need to follow suit. Like this piecemeal approach is not cost effective, and it's a headache. So this sort of streamlining, making sure all of the input materials can be sourced by the mRNA manufacturer is really key. So I want that to continue. The other thing I would say is it's also about flavor management. So as we've seen that it lends itself to platformability, right? But there's also the concept of, well, then how do we streamline that with the regulators? So as we have platforms and they're being targeted to generally the same sorts of systems in the body and stuff, how to reduce the cost by reducing the cost of pharmaceutical development, understanding what the CMC is going to be required and doing the minimal required rather than reinventing the wheel every step of the way, every time you do a new gene of interest. So that will also save a lot and help get these products through the clinic and actually helping people's lives a lot faster. So flavor management is going to become really important, especially in the rare disease space. Like we can't, we're already seeing this, like with gene therapy products, is if you're selling them, if you have to sell them for more than what people can pay, and like it doesn't work out. So we need to see this consolidation. We need to see this standardization of how we're going to get these regulatory packages through.